And hello, everyone. Welcome to DrugBank in Conversation with Professor Eran Segal. My name is Chris, and I'm the Scientific Support Lead at DrugBank. I'll be your moderator today. Today, we are very privileged to be discussing personalized medicine based on deep human phenotyping with Professor Segal from the Weizmann Institute. Professor Segal is a professor at the Department of Computer Science and Applied Mathematics, the Weizmann Institute of Science in Israel, heading a lab with a multidisciplinary team of computational biologists and experimental scientists in the area of computational and systems biology. This group has extensive experience in machine learning, computational biology, and analysis of heterogeneous high-throughput genomic data. His research focuses on microbiome, nutrition, genetics, and their effect on health and disease. His aim is to develop personalized medicine based on data from large-scale and deeply phenotyped human cohorts. So today, we'll be having a pretty standard format. So there'll be about 45 minutes for Professor Segal to present his work to us. We'll then have about 10 minutes for Q&A. If you have questions during the presentation, please put them in the chat bar. We'll have someone who will be monitoring that and collecting the questions. We'll try to get through as many as we can, but sometimes we don't quite manage. But we'll do our best. And then at the end, we'll have a couple of minutes for wrap-up. As a further note, this webinar will be recorded and will be shared with all the attendees and registrants via email in the next few days, so you should get a link to that. And now, without further ado, I will pass things over to Professor Segal, and we can get started.
Thank you very much, Chris, and thanks everybody for joining us. So let me share my screen. And very happy to be presenting you work that we've been doing well, for the past over a decade or so, and especially focused on work that we've been doing in the past 5 years on building a very large-scale and deeply phenotyped human cohort. As Chris mentioned, I'm a computer scientist by training, and as such, when we were thinking about what we can do to improve healthcare and human health, we've been thinking about how data could contribute. And I think in the field of biology and medicine in general, in my view, one of the major breakthroughs in the past several decades has been the publication of the human genome. So as you all know, in the year 2000, we got the first draft of the human genome. And if you look at what advances we've made, it's really been mind-blowing, because we went from knowing in the year 2000 about genetic variants involved in just a few diseases to 20-something years later, knowing about the involvement of hundreds, sometimes even thousands of genetic variants in virtually every single disease by genotyping millions of people. And it's also been shown that when genetic evidence is being brought in and used to support the targets that are being developed for drugs, that typically increases the success rate and thus lowers the cost of drug development by about 50%. But at the same time, while the human genome has really been transformative, we also know that the human genome is what we're born with. It doesn't change with life. And it doesn't take into account any environmental factors or nutrition, lifestyle, and so on and so forth, which we know are really critical for our well-being. And so for that reason, seeing what the human genome has done for biomedical research, we initiated what we call the Human Phenotype Project, where here the goal is to measure the genome, but in addition, measure everything that we can about people in very, very deep phenotyping, which I'll describe in a moment. And we believe that by phenotyping people very, very heavily, we'll be able to understand the well-being of people, understand trajectories of disease, be able to predict them ahead of time, and therefore be able to intervene and prevent them, as well as also use the data that we have to identify novel targets for drug developments and even further lower the cost, which is very prohibitive as you well know, for new drugs. And eventually this, we believe, will also lead up to personalized AI-based remote healthcare. So I mentioned the deep phenotyping that we do. So let me now describe what we actually measure. And this is really a key slide in this presentation, because this will tell you all of the data that we're collecting. And as I mentioned, the cohort that we have built so far is, at least at this point, not the biggest cohort in the world, but it is, as far as we know, the most broadly characterized, the most deeply phenotyped. So we built a website. People, by word of mouth, hear about this site here in Israel, about the study, and they can join online. Once they join, they fill out hundreds of questionnaires about their medical background, family history of disease, any medications they're taking, their current diagnoses, and they upload their entire electronic health records. So we also have all of their historical blood tests. They then come to a physical visit at a clinic that we set up at the Weizmann Institute. And in this clinic, they undergo, in about an hour and a half or 2 hours, they undergo a series of different tests. And what we measure, you can see here, are the various body measures. We call this anthropometrics. So blood pressure from lying down, standing up. We do hand grip strength. We do various other body measures. We also do blood tests on site so that we can standardize across people. We do various measurements of the cardiovascular system. This includes ECG, ABI, which measures the difference in blood pressure between the arm and the leg, and is also considered a gold standard in identifying blockage of arteries. We also record voice, which has a correlate to disease. Then we have various imaging data types, imaging modalities. So with DEXA, we look at full body composition and bone density. We use ultrasound to look at fat in the liver and to look at the carotids, also for cardiovascular health. We use OCT imaging to look at high resolution images of the retina, from which we can see blood vessels, their density, curvature, number, their shapes, and that's also considered to be a window to the heart and allowing us to assess cardiovascular health. Then maybe what makes the study most distinctive is all of the multi-omics testing that we do. We do a very large panel. The majority of multi-omics that can be done at high throughput and relatively low cost is what we apply in this study. And this includes the human genome, so whole genome sequencing. It includes looking at the microbiome, the collection of bacteria that live among us. We look at the vaginal microbiome as well as the gut microbiome. We do what we call metabolomics. So this is looking at thousands of different molecules in the blood, both targeted metabolomics as well as untargeted metabolomics, which we do by separating molecules according to their mass using a mass spectrometer. We do proteomics, which looks at levels of proteins in the blood. We do RNA sequencing in bulk because single cell RNA sequencing is not feasible for the size of the cohort that we have, but we do bulk RNA sequencing from immune cells in the blood. And we have a novel immunology assay whereby we can tell the entire infectious history of a person, all of the viruses that people were infected with, for which they still carry antibodies for. We do this by a phage display system that allows us to synthesize hundreds of thousands of antigens of our choice, which we can use to cover all of the known viruses. And then we go in with the antibodies of a person from the serum, get all of the phages that display antigens that get bound by antibodies, and we sequence them. And this is how we can do this assay. I should mention all of these multi-omics assays, with the exception of the human genetics and proteomics in-house, using automated robotic pipelines that we developed that allow us to do these at scale. We typically work with 96 or 384 well plates to run these assays, and we do them for a fraction of the cost compared to commercial list prices, sometimes fivefold, sometimes even tenfold cheaper than than what's available. And I should also mention, if anybody here also has cohorts or wants to profile multi-omic data sets, then we also have we can process that in the lab and then compare it to all the other data that we've been measuring. In addition to these multi-omics, we also use sensors that people go home with, so smartwatches to track physical activity, sleeping devices that for 3 nights with several different channels characterize sleep quality, something that was never assessed on a scale of thousands of people, and coupled with all this other information. And we use continuous glucose monitors, which for 2 weeks assess the glucose levels of each person every 15 minutes, allowing us to get a continuous profile of blood glucose of people. During those 2 weeks, people log all of their dietary intake. They're incentivized to do that because we can then tell them which foods and meals spiked their blood glucose levels and which did not. They also log all of their medications, and they track physical activity. So we can also couple that with the continuous glucose monitoring. And finally, at the bottom left, you can see the biobanking that we do. We have tens of thousands of biological samples from immune cells, from PBMCs, live cells that we store and that we can do future assays on, as well as serum and plasma. So this is the profile that we collect on each participant. We've so far profiled 12,000 people in Israel. The baseline cohort is a healthy cohort, but we also have several thousands of people with disease. And this is a longitudinally profiled cohort. Every year we track this cohort virtually, and every 2 years people come in physically to undergo all of the tests except for the human genome, of course, which does not change. Seeing the value of this data, which I'll spend the remainder of the talk on telling you about discoveries we've been making with this data, but seeing its value, hopefully you'll agree with me that there's a lot of value in this data. We are also in the process of significantly expanding this cohort to 100,000 people, which is our next milestone. And we hope to do that both by doubling the cohort in Israel, as well as opening and profiling this cohort, such a similar cohort in other countries. There's already 4 additional countries in which we're going to be doing these tests, which will increase ethnic, genetic, and cultural diversity. Now, if we think about the impact that such a cohort can have worldwide, I believe that this could really have tremendous potential. And I draw by analogy on the very impressive and amazing impact that a similar study has been having, and this is the UK Biobank. So this is a cohort of 500,000 people from the UK that started over 15 years ago and has been tracking people with a similar approach to ours, except that the profiling has been not as deep per person, but it is 500,000 people. And if you look at the impact that this project has made, it's really been incredible. So you can see just on publications in PubMed, over 8,000 publications have been using data from the UK Biobank in the past 12 years. It's been ramping up, as you can see on the graph, with over 2,000 publications based on the UK Biobank just last year. And overall, there's 30,000 researchers from 100 countries working on this data, and major investments by pharmaceutical companies to increase the offering, increase the data that exists here. And it really changed the face of how we do biomedical research. The vast majority of people are now using this type of data. In comparison, our cohort is smaller, but as I mentioned, it's also growing. But it does have several features that are not in the UK Biobank, so it's very complementary. It will be the first cohort that will be international and span more than one country. And then we have all this list of additional tests that you can see here: the microbiome, the RNA sequencing, the immune profiling, the metabolomics, sleep monitoring, continuous glucose monitoring, and the liver ultrasound, which are not part of the UK Biobank, and therefore make our data set highly complementary. I will also mention that now we are making this data publicly available. It will be available free for academics to use on a cloud-based platform that we developed that makes this data standardized, cleaned up, and so on. And for commercial entities, you can get access for a fee. And if you're interested, there is actually pheno.ai. You can go online, or you can email me and I can connect you. That's where this cloud-based platform is in, and you can get access to this data set. You will not be able to download the data, but you'll be able to work on a secure cloud-based environment with any analyses and any software that you want to use. You can even bring your own data, which nobody else will be able to see, to couple with the analysis here. And so anybody that this is of interest to, please contact either pheno.ai or contact me and I can connect you. So in the remainder of the talk, what I want to do is tell you about discoveries and works and research projects that we have been doing on this. There's numerous such projects. We have dozens of data scientists working on analyzing this data. We also realize that even a group of dozens of data scientists will only be able to scratch the surface of what could be discovered from such a data set, which is why we are making it available so that we can increase the impact of this entire endeavor. But what I'll do now is walk you through at a very high level so that I can cover and touch upon multiple different discoveries, several of the different discoveries that we've been able to make. So the first project I want to start with is actually a project that we did that predated this the building of this cohort. It was the cohort that led to the idea to build an even larger cohort, and this was a cohort in which we mainly profiled blood tests, microbiome, and continuous glucose monitors in people. And what we did, we published in 2015. We recruited 1,000 participants, them to these CGMs. The people logged all of their dietary intake, and analyzing the data, we reached three main discoveries. The first, as you can see in this graph here, we showed that when you give people exactly the same food, in this case 4 slices of bread, they actually had very reproducible blood glucose responses on the mornings of 2 different days, which you can see here from the 2 lines of the same color from the same participant. But you can see that across people, we saw huge variability in the response, where some individuals, they ate 4 slices of bread. We could not detect any change in their blood glucose levels, whereas others had a higher response and others had an even higher response. So this showed for the first time with actual data that people have personalized blood glucose responses to food. That was the first main discovery. The second was we were able to take microbiome data and clinical features about people and develop an algorithm that could fairly accurately predict these personalized responses to food. The axes here show the measured versus the predicted response, the area under the glucose curve after you eat a meal. There's 50,000 meals from 1,000 participants represented in this plot, and you can see that there's a fairly good correspondence. And finally, we were able to show that based on this algorithm, we could take individuals who are pre-diabetic, who normally have these huge spikes in glucose levels, and we could tailor a diet specifically to them, sometimes including surprising foods like ice cream, chocolate, pizza, and so on, not during the entire week, but on occasions. And prescribing these personalized diets, we could show that the glucose profile during an entire week, which is what's shown here, could be normalized in the same person eating exactly the same amount of calories, but choosing foods according to the algorithm. So this study we published in 2015, and since then, we've done a randomized clinical trial to see whether these diets have a long-term health benefit and impact. And so this is a study we concluded a couple of years ago on 200 people with prediabetes. we randomized them into either a standard of care diet, Mediterranean diet, or our algorithm-based diet. We then, for half a year, we gave them one of these two diets, depending on which group they were randomized into. And you can see here that taking a person who is pre-diabetic, and this is now glucose monitoring for an entire month, we could fully balance their blood sugar levels once we put them on the algorithm diet. This is anecdotally one person. And if we look statistically at the findings of this paper, you can see that by our primary outcome, which was changes in hemoglobin A1C, the average glucose levels over 3 months, we could see that the algorithm diet shown here in green had double the effect compared to the standard of care diet, which had some reduction after 3 and 6 months, but was half of what we got compared to the algorithm diet. And the algorithm diet further reduced many spikes in glucose levels, and it improved virtually all other metabolic parameters, including blood pressure, liver enzymes, cholesterol levels, fat lipids in the blood, and so on. So this for us was very gratifying. It concluded a decade of research in the lab, going from an idea to development of an algorithm for personalized nutrition, all the way to a randomized clinical trial that showed health benefits in both people with prediabetes and people with type 2 diabetes. Continuing on the theme of the microbiome, we have a very extensive research program looking into the microbiome, and specifically into finding what we call data-driven probiotics for trying to treat several different conditions. The computational pipeline that we developed at a high level, it starts from identifying bacteria that we believe are causative for some conditions. Once we identify those, and I'll go into in a moment into the computational discovery pipeline, but once we identify the relevant bacterial strains, we go back to participants who harbor the strains that we think are beneficial. We extract them, and then in a GMP facility, we grow them into a consortium and hope to go into clinical trials to show the efficacy that these bacteria have. The two first indications we're going to be focusing on are type 2 diabetes and weight loss. But we've been working on many indications, and for almost all of them, we find bacteria that we believe not only are correlative, but also are causative and are trying to follow the same approach. So here, very briefly, the computational pipeline, because we have so many participants, actually in the microbiome, our data set is by far the largest as a single cohort that exists worldwide. We map using the sequencing reads that we take from microbiome samples, from gut microbiome samples. We map all of the sequencing reads onto bacterial genomes. Once we have that mapping, we record, as you can see here in blue, the genetic variation that we see across samples at every position across all bacterial species. And we then link genetic variation that we see to phenotypic variation. So in this illustration here, we see that these three individuals, if you can see this, the first one has an A at the first position, the other two have a T at the highlighted position. The BMI of the first participant is high, the BMI of the other participants is lower. So this would constitute an association. This is, of course, an illustration. In practice, we do this on all of the data that we have on millions of different bacterial SNP across thousands of bacterial species. And what we have is, as a result for doing this for BMI, is a Manhattan plot, which I'm sure you've seen for human genetics. But here, instead of the x-axis being human chromosomes, the x-axis is now bacterial species, and every dot here represents a single position in a bacterial genome that we found a statistical significant association with BMI in this case. The y-axis is the p-value. You can see very low p-values, very significant associations. But even more interesting than that is, I think, the effect size. So this is the same data plotted. Every dot is still a SNP in a bacterial genome, but now the x-axis is the amount of BMI points that are statistically explained by every single SNP here. And you can see some SNPs in bacteria explaining 1 or even 2 points of BMI. This is a huge effect. It can amount to 10 or 15 pounds of body weight. And it's a huge effect. It's bigger than the largest effects that were seen in human genetics, even though the data set that we have on microbiome is much smaller. To me, this is not surprising because we think of the microbiome as an environmental factor, and as such, it's influenced by many environmental factors, and we know that obesity is driven partly by genetics, but even to a larger extent by our eating patterns, lifestyle, and many other environmental factors. Here's what it looks like at the strain level. So this is one bacterial species, and for simplicity, let's call this the blue and the green strain that we identified. You can see the blue strain is associated with people with lower BMI compared to the green strain associated with people with a higher BMI. This one explains 2 points of BMI. But not only that, when we look at what happens to people 2 years later, we see that people who harbored the blue strain associated with lower BMI did not gain weight, whereas people harboring the green strain on average gained another 1 point of BMI, and could be another 5 or 10 pounds of body weight. So these could be really big effect sizes. So we are in the process now of basically identifying these bacteria, and for example, going to individuals who harbor this blue strain, trying to get a stool sample from them, isolating the relevant bacteria, and then going into eventually clinical trials to test the efficacy of this, in this case for weight loss. Of course, we identify multiple different bacterial strains, so here's another one. And as I mentioned, we also find this for multiple different indications. I want to share with you also another work that we've done as a proof of principle on just 9 patients, but showing that bacterial interventions, in this case, in a coarse form, in the form of fecal microbiome transplants, could actually help ameliorate symptoms of a disease, in this case, atopic dermatitis. And so what we did here was following placebo capsules with no fecal material. We gave every 2 weeks a dosage of fecal material from a healthy donor. And you can see after the placebo, very clearly clinical manifestations of this skin disease. And after 2 dosings of FMT, we see clearing of these clinical symptoms. Here's another patient, and clearing even many weeks after we stopped dosing the patients with the fecal material. Our clinicians quantified this into an average reduction of 70% in clinical symptoms of the patients. And when we looked at under the hood on the microbiome, we saw that when we started, obviously donors whose samples here are shown in ellipses are quite far away from the recipients, from the patients who are represented by squares here. Once we do the placebo, we don't expect and we don't see any change in the microbiome of the patients. But once we begin the FMT, we see a major shift in the microbiome of the patients. They become, with the exception maybe of one participant, they become very similar to the microbiome of their donor, not identical, but they undergo a major shift in their microbiome and become much more similar. And so we believe that it is this microbiome transfer that underlies the actual improvement in clinical symptoms. Another area that we work heavily on, I mentioned this before as one of the data types that we measure is blood metabolites. Here, we took advantage of the fact that we have very broad profiling of people and we measure many different categories on them to ask a very fundamental question, which is what determines levels of blood metabolites? Is it more, for each metabolite we wanted to ask, is it driven by diet, genetics, risk factors, microbiome, and so on. And so using all of these different data types, we were able to show, we published this 3 years ago, that for 90% of the metabolites, we could find a factor that explains them well, that may be the factor that determines and modulates their levels. And you can see here that on the left, we have metabolites that seem to be driven by gut bacteria. In the center, we have metabolites that seem to be driven by diet. A simple positive example here is levels of caffeine at the bottom here are predicted by diet. Specifically, the dominant features is the amount of coffee that people drink, not surprisingly, is a major determinant of their levels of caffeine. But then there were many other metabolites that were explained by diet that were much less obvious. And then we have other metabolites that seem to be mostly driven by genetics. So the reason why we think this work is fundamental and important is because we know that circulating metabolites in our blood could be very key for our health. And therefore, if we identify metabolites that are important for disease, the next thing that we'll want to do is to modulate their levels. And so a prerequisite to that is knowing what factors actually control and determine these levels so that we could intervene and change them and then affect and modulate the metabolite. But having this understanding and having this mapping between factors that determine levels of different metabolites allows us to go and then shed light on different disease cohorts. And I want to give you one example work that we published a couple of years ago based on this association of metabolites and determining factors. Here we analyzed heart disease patients, people with acute coronary syndrome. And we showed that you can take even a single patient and go into our healthy cohort and identify a matched reference cohort for that single patient. For example, if we have a 60-year-old male who underwent a heart attack, we would extract 60-year-old males who are healthy in our cohort. And that cohort would be the correct and best reference group that we can provide for analyzing our single patient. And once we have that, we can build a norm for each metabolite based on this personalized reference cohort for this person. So what is the distribution of levels of this metabolite, of each metabolite in not just a healthy cohort, but in the healthy cohort that's matched by whatever we want to match, let's say age and gender, to our patient. And then we can see where the levels of that metabolite fall for our patient. In this case, you can see that it falls at a low value outside of the distribution of the expected values based on the right reference cohort for this participant. And we can quantify that by a z-score, by the number of standard deviations that it's away. We can, of course, do that for all of the different metabolites, and we end up with a list of metabolites that seem to be abnormal, that seem to deviate in this patient as compared to his personalized reference matched control of healthy individuals. And once we have this profile of all of the list of metabolites that deviate in this person, we can go back to the database I mentioned before of determining factors for metabolites and ask whether the metabolites that deviate in our patient, are they metabolites that are driven by diet, microbiome, genetics, or other traditional risk factors? And when we do that, we see that two patients that present at the clinic with the same conditions, they both underwent a heart attack. When we look at the metabolites that deviate, in one participant, it could be that his deviating metabolites are mostly driven by genetics, whereas in somebody else, they could be driven by diet, and in yet another patient, they could be driven by the microbiome. And so we believe that this also explains why when we provide patients with the same medication, they don't all respond to it, and it's because the pathophysiology of disease, even though the endpoint is the same, may be very different across people. And so this is an example of how we can identify what are the drivers potentially of disease and where we need to intervene in order to assist people. In mentioning, in passing, I'll mention using multiple layers, we were able to look at multiple sclerosis and identify, in this case, a specific pathway that produces indole propionate. This is a metabolite that's known to protect neurons from oxidative damage, and we identified that both the actual metabolite in the blood as well as bacteria that produce a precursor to this metabolite are lower in MS patients as compared to controls. And because of what we know about this pathway, this may suggest a pathway in which, if we intervene and increase its levels, might actually help MS patients. Here's another work that we've done on inflammatory bowel disease, or IBD, where here, again, we were able to make use of the very large cohort that we have to take just 100 IBD patients, but match them by age and gender to over 1,400 different healthy controls, and then do a very robust analysis, which in this case identified microbiome biomarkers that separate IBD patients from healthy controls. And the final work I want to tell you about is work that we've been doing on human aging, or what we call biological aging. So here we took our metabolomics data, and using only the metabolomics data, we were able to build, as you can see here, a machine learning algorithm that fairly accurately predicts the chronological age of people. But you can see that this clock is far from perfect. There's people who have a higher predicted metabolomic age, if you will, compared to their actual chronological age, and conversely, individuals who have a lower predicted metabolomic age according to the model. In other words, their metabolites, when the model looks at them, resemble somebody who's younger. So we then asked whether differences between these populations actually carry some meaning, and indeed, we saw that if you compare people who, according to the model, have a higher metabolomic age compared to people who have a lower metabolomic age, they are indeed in worse health condition. So people with a higher metabolomic age have higher BMI, higher blood lipids, triglycerides, higher hemoglobin A1C, blood pressure, and cholesterol and liver enzymes. So they are indeed in worse health condition, and so we believe that therefore this this model, this biological clock, actually carries a clinical meaning. But what I've showed you is only based on the metabolomic data. Of course, we have multiple different types of data. And so we took all of our data, we partitioned it into 14 different body systems. So we grouped together different measurements that we have on the same body system. And for each of them, we developed this chronological or biological clock. All of them were significant, so explaining a significant fraction of chronological age. And in this study, we made three main discoveries. The first is that we found that these clocks were largely independent, meaning that if one system of your body ages faster, it doesn't necessarily mean that another system of your body will age faster. This was actually quite a surprising result. The second was that we were able to show that these clocks, like I showed you for the metabolomic age, they actually have clinical meaning, because we found that if you have a higher age, according to one of the clocks, you will be at risk for developing a disease, but actually not only just a disease, but a disease related to the body system on which the clock was developed. For example, the clock for insulin resistance, if you have a higher biological age there, you're at higher risk for developing diabetes, obviously related to this body system. And the final discovery was a marked difference between males and females, where we saw that males age relatively linearly, whereas in females, we saw this jump right on the age of 50, which we could link to menopause. And so comparing women of the same chronological age, but pre versus post menopause, we saw multiple different clocks on which the women became older on. So women who pass menopause undergo significant changes in these various body systems, and they have a higher biological age. And we think that this could be a diagnostic to also know when to intervene in women, perhaps with hormonal therapy that may delay their onset of menopause, and that's been shown to, in the long term, reduce diseases like cardiovascular disease. And the final thing I want to mention is that in this day and age, we are also using AI to develop models that integrate together multiple different types of data. For example, building representations of both the retina scans that we have and the images that we have from the carotids. Both are looking at different measures of the cardiovascular system, and we're using these AI models to build joint representations that hopefully would be better at capturing heart conditions. Similarly, integrating together diet and continuous glucose monitoring, we show that using only diet, we can then reconstruct glucose patterns of individuals. And so in closing, I hope I convince you about the value of this data. And I mentioned that because of this value, we're now in the process of significantly expanding this cohort to go from 12,000 to 100,000, to expand to different geographies that would allow us to sample more cultures, more ethnic backgrounds, and so on. And also to expand the list of diseases that we are profiling, as well as go into multiple other measurements. Even though we are very broad in what we measure, we don't measure everything. And I should mention, we've developed everything in-house, from recruiting the cohorts to the actual physical clinic in which the participants arrive and get profiled, to the actual biological lab that processes biological samples and produces the multi-omics data, including some proprietary assays, and then all the way to our core expertise, which is in the data analysis. We also are interested in collaboration. So if you have a cohort that you want to profile, we have systems that allow us to manage and profile these cohorts, and the ability to carry out a multi-omics on them. So please reach out either on the website to pheno.ai or to me personally, and I can connect you, and we'd be very interested in collaborating. We have multiple such collaborations that led to assemblies of other cohorts in 4 additional countries. And overall, I believe that this is really what will take us to be able to build the next generation of precision medicine, and really would be these types of cohorts would really be a moonshot to improving human health. And with that, I'd like to end and thank you, and just put the acknowledgement slide of all the people in my lab that led this work and discoveries. And maybe I'll stop here and be happy to answer any questions that you have. Thank you.
Yes, thank you very much. That was an absolutely fascinating presentation. So we'll now open the floor for a little bit of Q&A. It looks like we have pretty much exactly 15 minutes as we hoped for. So again, if you have questions, please put them in the chat. We'll be answering them in the order in which they arrive. Okay, so we already have a question here, and it is, have you looked at any drivers of chronic pain? And then as a second part, what are your thoughts on human stem cell-based assays?
Yeah, so we haven't looked specifically on drivers of chronic pain, but we do have many participants who actually reported chronic pain. So now this is actually a great example of some trait that can now be looked at and analyzed with all of this very comprehensive profile that we have of people. And stem cell based assays, yes, I think those are the types of one of many types of assays. by which we can learn a lot about cellular state and interactions between genes and various cellular phenotypes, which could then be taken. This type of information could then be taken and integrated into analyses that span multiple layers of data, like what we have here.
Fantastic. Okay, so as we wait for more questions to roll in, I had a question of my own, if you don't mind. So I know that one of the kind of criticisms, if you will, of a lot of the genome-wide association studies that have been performed historically has been a real bias towards, individual populations and a prevalence of Europeans over other kinds of ethnicities. And I'm curious what your thoughts are on how any kind of a bias in your current cohort may affect what you do or even don't find, especially because you have so many more types of data than just the genomic data.
Yeah, no, that's a very valid and fair question. So I'll say two things about that. The first is, so in general, yes, I mean, I agree, but I think Israel is actually considered, where we profiled all the participants so far, to be quite a diverse place, genetically speaking, because only several decades ago we had people coming in from and migrating into Israel from different parts of the world. We have the Ashkenazi Jews, the Sephardi Jews, so they're very different genetically. So it is considered to be a genetically diverse place and population, also culturally. But of course, it's a very small fraction of all the diversity that we have worldwide, which is why my second response to this is actually the main reason why we're expanding to additional countries, exactly in order to profile different cultures, ethnicities, genetic backgrounds, lifestyles, and so on. And I think really by building international cohorts that span these countries, we'll be able to get to a more generalized representation. And the first countries that we're going into are actually countries mostly not of European descent, which would actually profile into largely untapped territory in terms of what has been profiled so far.
Oh, fantastic! Yeah. Another question I had was, I was curious if you'd ever looked into. So I'm not an expert in the microbiome, but I'm curious to know if you have any insights from your data about how things like an individual's genetics and diet then play a role in shaping what kind of diversity they have in their microbiome, or if that isn't something that you've had a chance to look into.
Yes, absolutely. So what I will say is what we are finding is actually it's a somewhat complex answer, but I think it has a simple principle. So we see that people, relatively speaking, have a very stable microbiome while they're adults. So your microbiome probably gets shaped mostly during childhood, and then it remains quite stable. Having said that, however, we do see that when people change their diet, they actually undergo changes in their microbiome. And the way that you can kind of merge these two, what seems conflicting views, is that on the one hand, your microbiome is stable. Most of the bugs that you acquired will stay with you. However, you can still experience some changes that could also have very significant impact, but these changes will typically be much smaller than the changes that you see across different people. In other words, your microbiome will change, but it'll still be more similar to what your microbiome was, say, a year ago before you did the intervention or dietary change. But that change could still really have profound functional impact. And that's kind of what we are seeing. So diet definitely shapes a big deal, a big part of the microbiome, but a lot of it also remains stable during adulthood.
Okay. Yeah, that's very interesting. Okay, we have one individual here who is interested in the Pheno.ai platform. So I would suggest maybe getting in touch with Professor Segal after this to learn more about that.
Yeah, please do.
Okay, there's another question here. What is the current status of HPP? I assume that's the Human Phenotype Project, in applications to psychiatric or behavioral health, likewise for a general umbrella of orphan diseases?
Yeah, so we haven't looked so much on psychiatric and behavioral health. This is actually some of the assays that and measures that we will be adding to the HPP. And likewise, orphan diseases, a cohort that I would say here our cohort is probably too small for that. So 12,000 people that is not targeted at a specific orphan disease, we probably won't have enough statistical power. So I think this data set is good for many things, and there are some things where it's limited in, and this is definitely one of them. It is I think its advantages are for the more prevalent diseases that you would already see dozens of individuals in, even with a cohort of 12,000 people.
Mm-hmm. Yeah, I think that's a general issue that a lot of people face with orphan diseases is just sample size. And even if you have a targeted cohort, I think it still can be very difficult depending on the prevalence of the disease in the population.
Yeah.
Okay, we have another question here. Have you measured effects on patients that, say, have had steroids used to treat medical conditions as a child, such as asthma? Any insight into how that affects them and their microbiome, either as a child or as an adult?
So this we definitely have measured this. So part of the questionnaires that we have, people report their medical history, so we would know if they had asthma as a child or other conditions. We haven't specifically analyzed the data for this purpose, just because we've been focusing on other things. But I think this would be very interesting. And like I said, the data you can access the data either as an academic for free or for a fee for commercial entities to ask these and many other questions that we haven't asked so far.
Yes, definitely. Okay. There's no more questions. I had one final question, if I may. So I think regardless of the type of model you're building, whether it's statistical, machine learning, finding the right parameters or the right features, I think, is always challenging. And especially in your case, you almost have too much data to work with. So I'm curious if you could share a little bit on how you've managed to deal with that particular problem and find the right features to build algorithms that actually have good predictive power.
Yeah, so that's a great question, and really the art of data science and machine learning. And we address this in many different ways that are standard in machine learning, basically compressing the feature space, finding features that have a high signal to noise, using prior knowledge to prioritize what features we look at, and always, of course, evaluating ourselves on people that we hold out, that we have not used as part of the training. So a lot of the toolbox of in general machine learning, and also there's many correlations between many different features that we measure. Many things are correlated in obviously in the human body, and so in effect, even though there are many different parameters, many of them are correlated, and and therefore effectively there's actually a smaller actual space of features. But I'll say this is definitely a challenge, which is why we're also working to increase the number of people so that we'll be able to have a better ratio of parameters to participants.
Yeah, definitely. Okay, so I think with that, we'll start to wrap things up for today. I'd like to thank everyone for their attendance and for their insightful questions. As we mentioned, this webinar is being recorded and it will be shared with all of the attendees and the registrants via email in the next few days. If you're interested in learning more about what you've seen today, you can follow Professor Segal on Twitter, on LinkedIn. He has a lab website. And then, of course, as he mentioned, there's the pheno.ai platform. And if you'd like more information on that, you can reach out to him afterwards as well. For more resources and webinars like this, please feel free to follow DrugBank on LinkedIn or visit drugbank.com. We hold these on a regular basis, and I think we're just getting better and better over time. And then finally, I'd just like to once again thank our speaker for his time, for his insight. I think that today's session was incredibly interesting. I know that I personally learned a lot, and I'm sure that everyone in the audience did as well. So yeah, thank you very much for taking the time to be with us today.
Thank you for having me. Great.
All right, and with that,
Thank you.
we will end. Thank you, everyone.
Thank you.